So the topic for today is the detail of generation and conduction of the nerve impulse. So nerve impulse is a wave of electrochemical disturbance that passes along the components of the nervous system during the conduction of an excitation. So what is excitation here? It is the impulse basically. So this impulse is supposed to be a wave of some electrochemical difference. This disturbance is because of the movement of the ions we learned from the NCERT in the last class. So when these ions move across, this causes disturbance in the polarity. So there is change in the charges on the outer surface and the inner surface of the membrane, and then this develops a small local current which causes the movement of impulse from one place to the other place, and which is called as the conduction of the nerve impulse. So the excitation is produced in response to the stimulus. So whenever action potential it develops because of a response to the receiving of a stimulus. Now the velocity of this conduction is variable. It is 10 to 20 meter per second in the case of the autonomic neural system, and it is 100 to 120 meter per second in the case of the somatic neural system. So just need to remember this, that the speed is faster in the case of the somatic. That's why you must have seen the muscle coordination working and all is faster in the case of the somatic neural system. The synaptic delay, now there is a delay in this process because the information has to pass from the neuron and then it passes through the synapse. So because there is a synapse will never generally don't have the electrical synapses in the case of human, because the synapse, you have learned even in the 10th class, it carries an chemical. So this release of chemical from the vesicle requires time, and then this chemical will be released into the synaptic cleft, that gap in between the synapse, and then it is picked up by the next neuron. So this whole process causes a slight delay in the process, and that is called as the synaptic delay. This delay is because of the release and activity of the neurotransmitter, which consumes some amount of time, and the time consumption is 0.3 to 0.5 milliseconds. So this much time is consumed during the release of these acetylcholine and then receiving of the acetylcholine by the next neuron. And then there is a refractory period also. What is refractory period? We all know once a nervous system or a neuron is supposed to receive a signal, after receiving signal, we cannot simply put another signal or stimulus to it. It needs to readjust. So that readjustment timing is called as the refractory period, and this period is 0.01 second, that means 1 millisecond. So this is the period during which the neuron is supposed to undergo the rearrangement or adjustment so that it can receive next stimulus. Now, as per the NCERT last time we have learned, the membrane potentials are of two types, resting potential and the action potential. Now the resting potential is minus 70 millivolt. It can range from minus 40 to minus 90, and it is the membrane potential of a neuron which is in resting situation. Now why we put it into negative value? Because the outer surface, which is the extracellular fluid, it carries the positive charge relative to the interior of the neuron. So inside it is negatively charged and outside it is positively charged, as we have already covered in the last class in detail from the NCERT. The action potential is written as the positive sign and 30 millivolt, and it can range from plus 20 to plus 60, so that is the range for it. And the membrane potential, this membrane potential is the potential of a neuron which is in action or is in excited mode, and the positive value indicates that the interior of the neuron has more positive charge compared to the extracellular fluid, as I explained in the last class, if you remember from the NCERT, that when the sodium ions channel they open, they start moving in, and as they move inside, it becomes positive inside compared to the outside. So that is called as the action potential development. The change in the membrane potential occurs due to the transfer of ions across the membrane through the ion channel. So there are specific ion channels through which the ions start moving across the membrane, and this causes the change in the potential. So from the resting potential it turns into the action potential, and the change in the membrane potential occurred due to the transfer of the ions across the membrane through the ion channels that are transmembrane proteins. So these channels are basically the transmembrane protein. So if you link this with your the fluid mosaic model, we have already covered this in the cell structure. If you remember, in the 8th chapter we have already done that there are proteins which span throughout the width of the membrane, and they act like the tunnels, or also called as the transmembrane proteins. And these proteins are basically gated. That means there will be a gate, and this gate only opens for the ions to move across when there is a particular stimulus applied. And the gates only open in response to a stimulus, and that stimulus can be voltage, that stimulus can be mechanical pressure, or the presence of chemical. It could be anything. Anyway, the gates may open, and once the gates open, for a brief period of time the ions will start moving across. So this will cause the change in the concentration of ions, and this will cause the action potential to develop, because now the polarities or charges have changed.